Regulation of glycolysis and gluconeogenesis, the role of catalase, by Chrissy Wilhelm. Today we will be able to understand glycolysis and gluconeogenesis regulation, learn atalase's dual role, compare atalase's isoforms, and analyze transcriptional regulation of atalase B. Glycolysis is the central pathway of glucose metabolism, breaking down one molecule of glucose into two molecules of pyruvate while generating ATP and NADH. This pathway can be divided into two main stages, the investment phase and the payoff phase. In the investment phase, the cell uses energy to phosphorylate glucose and prepare it for cleavage. Two ATP molecules are consumed in this stage. One when glucose is phosphorylated to glucose 6-phosphate by hexokinase, and another when fructose 6-phosphate is phosphorylated to fructose 1,6-biphosphate, or phosphofructokinase, which is PFK1. This phase essentially primes the glucose molecule for efficient energy extraction in the later steps. The payoff phase follows producing 4 molecules of ATP and 2 molecules of NADH per glucose. After the 6-carbon sugar is split into 2 3-carbon intermediates, glyceraldehyde 3-phosphate undergoes a series of reactions that generate high-energy products. Importantly, pyruvate kinase catalyzes the final step of glycolysis, producing pyruvate and ATP. Because 2 of the glyceraldehyde 3-phosphate molecules are processed per glucose, the net yield of glycolysis is 2 ATP and 2 NADH, making it a key energy-producing process in both aerobic and anaerobic conditions. Regulation of glycolysis is critical to ensure energy production matches the cell's needs, and this is achieved primarily through the 3 irreversible steps. There's the hexokinase/glucokinase regulates glucose entry into the pathway, ensuring glucose is phosphorylated and trapped in the cell. The PFK1 is the main control point, often called the committed step of glycolysis, because it is highly regulated by energy signals such as ATP, AMP, and citrate. Finally, the pyruvate kinase regulates the final ATP-producing steps and is also influenced by the energy status and hormonal signals. Together, the investment and payoff phase of glycolysis illustrates the balance between energy use and production, while the key regulatory enzymes ensure that glycolysis is only active when the cell requires ATP or a biosynthetic intermediate. This fine-tuned control allows glycolysis to integrate with other metabolic pathways such as gluconeogenesis and the citric acid cycle, maintaining overall energy homeostasis. Gluconeogenesis is the metabolic pathway that generates glucose from the non-carbohydrate precursors, such as lactate, glycerol, and certain amino acids. It primarily occurs in the liver and to a lesser extent in the kidney. While gluconeogenesis shares several reversible steps with glycolysis, it differs at 3 irreversible steps that must be bypassed using unique enzymes. These bypasses ensure that glucose synthesis is thermodynamically favorable and prevents a futile cycle where glycolysis and gluconeogenesis would occur simultaneously. The first major bypass occurs at the conversion of pyruvate to phosphonyl pyruvate in glycolysis. The pyruvate kinase irreversibly converts PEP to pyruvate, but glyconeogenesis uses a 2-step process involving the pyruvate carboxylase to form oxaloacetate, followed by the phosphopyruvate carboxyl to generate PEP. This step is tightly regulated and requires energy in the form of ATP and GTP, highlighting the energetic cost of glucose synthesis compared to glycolysis. Another key regulatory point is fructose 1,6-biphosphate, which catalyzes the conversion of fructose 1,6-biphosphate to fructose 6-phosphate. This step bypasses the irreversible PFK1 step in glycolysis and is sensitive to allosteric regulation by molecules such as AMP and fructose 2,6-biphosphate. In contrast to PFK1, which is activated by the AMP to promote glycolysis when energy is low, the fructose 1,6-biphosphate is inhibited by the AMP to prevent gluconeogenesis under energy-deficient conditions. Overall, gluconeogenesis represents a carefully controlled pathway that complements glycolysis, ensuring glucose availability during fast or high-energy demands. The unique enzymes at irreversible steps combined with tight regulation allow the cell to maintain blood glucose levels and energy homeostasis. This coordination prevents conflict between energy production and glucose synthesis, illustrating the intricate balance of the metabolic pathways in the body. Adalase is a key enzyme that functions in both glycolysis and gluconeogenesis by catalyzing the reversible cleavage of fructose 1,6-biphosphate, or F1,6BP, into 3 carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. In glycolysis, this reaction occurs in the forward direction, breaking down F1,6BP derived from glucose into intermediates that can be further metabolized to generate ATP and NADH. This step is central because it splits the 6-carbon sugars into 2 3-carbon units, enabling the subsequent payoff phase of glycolysis where energy is harvested. In gluconeogenesis, adalase operates in the reverse direction, condensing G3P and DHAP to form F1,6BP, which then progresses toward glucose synthesis. The reversibility of adalase makes it a versatile enzyme, allowing the cell to interconvert intermediates depending on metabolic needs. Because this reaction is near equilibrium, the directionality is largely determined by substrate and product concentrations, enabling smooth coordination between glycolysis and gluconeogenesis. This adalase serves as a critical node connecting energy production and glucose biosynthesis, highlighting its importance in maintaining cellular and systematic metabolic balance. Adalase exists in 3 different isoforms, Form A, Form B, and Form C, each with distinct tissue distributions and functional roles. Adalase A is primarily found in muscles, red blood cells, and fetal tissue, where it plays a major role in glycolysis. In these tissues, rapid ATP production is critical for energy-demanding processes such as muscle contraction and maintaining red blood cell function. The high expression of adalase A ensures efficient cleavage of fructose 1,6-biphosphate to provide glyceraldehyde 3-phosphate and dihydroxyacetone phosphate for continued glycolytic flux. Adalase B is expressed mainly in the liver, kidney, and small intestine, and is essential for both gluconeogenesis and fructose metabolism. In the liver, adalase B facilitates glucose production from non-carbohydrate precursors and is critical in processing dietary fructose. Deficiencies of adylase B leads to hereditary fructose intolerance, a condition in which fructose 1-phosphate accumulates, causing hypoglycemia and liver toxicity. Its expression in the kidney and small intestine supports glucose homeostasis and fructose processing in these tissues, highlighting its versatile metabolic role. Adylase C is predominantly found in the brain and nervous tissue, where energy metabolism is tightly regulated to support neuronal activity. Neurons rely on glycolysis for rapid ATP production, and adylase C ensures an adequate supply of those triose phosphates for energy generation. The tissue-specific distribution of these isoforms illustrates how the body tailors enzyme expression to meet the unique metabolic demands of different organs, enabling efficient energy production and glucose regulation across tissues. Adylase B plays a critical role in the liver, kidney, and small intestine by catalyzing the reversible cleavage of fructose 1-phosphate into glyceraldehyde and dihydroxyacetone phosphate, which can then enter glycolysis and gluconeogenesis. This enzyme is especially important for fructose metabolism, allowing the liver to convert dietary fructose into intermediates that contribute to glucose production or energy generation. Because the liver is central to maintaining blood glucose levels, adylase B is essential for proper glucose and energy homeostasis, particularly after meals containing fructose or sucrose. Deficiency of adylase B results in hereditary fructose intolerance, or HFI, a metabolic disorder characterized by the accumulation of fructose 1-phosphate in the liver, kidney, and small intestine. This accumulation depletes the phosphate and ATP, inhibiting glycolysis and gluconeogenesis, and leading to severe hypoglycemia, vomiting, and potential liver damage when fructose is ingested. HFI highlights the enzyme's clinical significance and demonstrates how proper regulation of adylase B is critical for safe and efficient glucose and fructose metabolism in humans. The expression of adylase B is tightly regulated at the transcription level to ensure proper liver glucose and fructose metabolism. Hepatocyte nuclear factor 1, or HNF1, and HNF3 are liver-enriched transcription factors that bind to specific promoter regions of the metabolic genes, including adylase B, to enhance their transcription. These factors help coordinate the liver's metabolic response to nutritional status, ensuring that adylase B is expressed when dietary fructose or glucogenic precursors are available. By promoting adylase B expression, the HNF1 and HNF3 maintain efficient flux through glycolysis and gluconeogenesis and support overall glucose homeostasis. Another key regulator is the CEBP, which controls genes involved in energy metabolism and gluconeogenesis. The CEBP interacts with the adylase B promoter to adjust transcription in response to energy demands, such as fasting, when glucogenic activity needs to increase. Additionally, the DBP, a circadian rhythm-associated transcription factor, influences adylase B expression in a time-dependent manner. DBP ensures that glucose metabolism aligns with daily feeding and fasting cycles, optimizing energy production and liver function. Together, these transcription factors integrate nutritional, energetic, and circadian signals to precisely regulate adylase B levels in the liver. Proper regulation of glycolysis gluconeogenesis, and adolase B expression is essential for maintaining glucose homeostasis in the body. When these pathways are misregulated, it can lead to serious metabolic consequences. For example, insufficient adolase B activity impairs the liver's ability to metabolize fructose and produce glucose, leading to the accumulation of fructose 1-phosphate, depletion of cellular phosphate, and inhibition of ATP generation. This disruption can cause hypoglycemia, vomiting, and liver toxicity, as seen in hereditary fructose intolerance. Similarly, if the gluconeogenesis is dysregulated, such as in diabetes, glucose production may become excessive or insufficient, contributing to hyperglycemia or hypoglycemia. These examples illustrate how tightly coordinated regulation of enzymes and transcription factors are necessary for metabolic balance. Adolase B's role in integrating glycolysis and gluconeogenesis and fructose metabolism highlights the importance of tissue-specific expression and transcriptional control. Misregulation at any level, whether through genetic mutations or metabolic dysfunction, can have systemic effects, emphasizing the clinical relevance of understanding these pathways. By studying such pathophysiology, students can appreciate how molecular mechanisms translate into real-world metabolic disorders and the importance of targeted therapies or dietary management. Glycolysis and gluconeogenesis are two central metabolic pathways that are closely interconnected but regulated in opposite directions to maintain energy and glucose homeostasis. Glycolysis breaks down glucose to generate ATP, whereas gluconeogenesis synthesizes glucose from non-carbohydrate precursors. Both pathways share reversible steps, but each has a unique irreversible reaction catalyzed by specific enzymes that are tightly regulated. Key regulatory enzymes in glycolysis include hexokinase, glucokinase, phosphofructase kinase 1, and pyruvate kinase, while gluconeogenesis relies on pyruvate, carboxylase, PepCK, and fructose 1,6-biphosphate. This reciprocal regulation ensures that energy production and glucose synthesis do not occur simultaneously, preventing futile cycling and conserving cellular resources. Adolase serves as a shared enzyme in both pathways, catalyzing the reversible cleavage or condensation of fructose 1,6-biphosphate. Despite this shared function, adolase exists in tissue-specific isoforms, A, B, and C, that reflect the unique metabolic needs of different organs. Adolase A predominates in muscle and red blood cells to protect glycolytic energy. B is expressed in the liver, kidney, and small intestine, where it plays a key role in both gluconeogenesis and fructose metabolism. The tissue-specific expression of these isoforms ensures that each organ can efficiently regulate energy production and glucose handling according to its physiological role. Adolase B in particular is tightly regulated at the transcriptional level by multiple factors including HNF1, HNF3, C, EBP, and DBP. These transcription factors respond to nutritional, hormonal, and circadian signals, adjusting adolase B expression to match the liver's metabolic demands. Proper regulation of adolase B is critical for maintaining glucose homeostasis and preventing metabolic disorders such as hereditary fructose intolerance or hypoglycemia. Overall, the integration of enzymatic activity, isoform specificity, and transcriptional control highlights the sophisticated coordination required for efficient energy metabolism and underscores the clinical relevance of these pathways. For more information, please check out my references. Thank you for watching.